everyone, and welcome to this webinar on EMBL-EBI's Job Dispatcher Service. And thank you for joining us today. My name is Nandana Masudanan, and I work with the Job Dispatcher team here at the EBI. So this webinar mainly aims to introduce you to our new website and demonstrate how to navigate through various sequence analysis tools. So those who are new to this, the Job Dispatcher team provide free access to a wide range of bioinformatics tools for sequence analysis. We have over 50 tools in Job Dispatcher now, including popular applications like Clustal Omega, BLAST, FastA, and EMBOSS suite of tools. These tools are run on EMBL-EBI's high-performance computing infrastructure, which ensure fast and reliable results. We also provide web services in addition to our web interface. These services are not only used by users like you, but also internally within EBI, like Ensemble, Genome, Uniprot. So these teams use our web services. To access our tools, you can either go to the EBI's main service page, which is ebi.ac.uk/services, where you can type in Job Dispatcher or JDispatcher. It will list our service, or you can directly go to our website. It is accessible from ebi.ac.uk/JDispatcher. So this is what our new website looks like. We have completely revamped the Job Dispatcher web pages to provide a more user-friendly experience. So this is the first time we have a single landing page that provides a comprehensive overview of all the tools and tool categories. So with this new website, you can quickly locate and easily access your results. So as you can see here, like you can provide, so if you want to retrieve your result, you can provide your job ID here and it will give you the result. And it's equally useful to our programmatic users if they wanted to visualize their result. You can only retrieve one job at a time and make sure that you provide a valid job ID. We store your results only for a week from the submission. So make sure that you download them if you need the output for later. And we also provide a job history. So you can access it from the recent jobs or your job lists section that lists all your recently submitted job. From the homepage, it lists only 5 recently submitted jobs. But if you click on more, you can see all your jobs that are submitted within the 7 days. It also provides the status of the job, whether it's running finished, and when you launch the job, you can also delete any unwanted listing from here. So as I mentioned earlier, the homepage lists and categorizes all the tools we provide. So we have several tool categories, for example, pairwise sequence alignment, that can be used to identify sequence patterns or mutations or regions of local or global similarity between 2 sequences. We have tools from EMBOSS like needle, stretcher, water matcher, etc. in pairwise. We also got a few FastA programs also in the pairwise sequence alignment. And we have a multiple sequence alignment, which has Clustal, K-Align, Muscle, T-Coffee, etc. So these tools can be used to find conserved sequence motifs that are preserved across multiple sequences. So these conserved motifs often play a critical role in protein or genes function. So they might be binding sites for other molecules, active sites for enzymatic reactions or regulatory regions that control gene expression. So if you're interested in conserved regions between several sequences, you can go for any of these multiple sequence alignment tool. We also provide sequence similarity search tools like BLAST, FASTA, and a few iterative search tools as well that allows you to search your query sequence against EBI's sequence libraries. So these tools helps to annotate genes or identify similar proteins. So we also have got protein functional analysis tools, tools for RNA analysis, and many other tool categories. Just to add that, we also got a service called DB Fetch, which stands for database fetch, that acts as a single point of access to retrieve various entries from different EBI's databases. So we have also started adding blogs and tweets about our recent activities, like announcing about our new dataset release, which is used by our similarity search tools. So from the tool category, like you can navigate to the respective category page. This is an example of pairwise sequence alignment. This page gives you a brief description about the tool, about each tool under that category, and that will help you to choose the right tool for your analysis. Say, for example, if you would like to compare homologous genes or proteins, say, for example, a human versus mouse, you can go for a global alignment, which do an end-to-end alignment. Whereas if you're looking for conserved patterns in two sequences, go for a local alignment that helps to find the local regions with highest level of similarity. While selecting a tool, consider the size of your sequence. Choose the appropriate tool that can deal with lengthy sequences or massive sequences. So it's recommended to read the tool description before using any of our tools. Sorry. So this is how our tool page looks like. So this is an example of Clustal Omega's tool page. First, you need to specify the type of your sequence, whether it's protein or a nucleotide. And there are options to select or adjust parameters based on your requirement. Make sure that you provide a valid input sequence in the tool's supported format. We support FASTA, EMBL, UniProt, and many other formats. And you can see the example formats by clicking on this, which will lead you to the documentation page, and list gives you the example sequence formats. Say, for example, if you are using MView, that requires an alignment result as an input. So make sure that you have provided the right format. We are no longer providing any individual help and documentation for each and every tool. But you can find the tool's parameter information by clicking or mouse over this information icon, which will give you details about what the parameter is. And I will walk you through the documentation page later in the session. And you can adjust the parameters and once before, we are not providing the notify by email option. Before we used to, but now we have that the list of jobs you can accessible from the homepage. So we are no longer providing the email address. Okay, so before submitting the analyze, we will perform an initial validation from our side. Say, for example, if you're trying two sequences at the first search box of a pairwise alignment tool, it shows an error. Like only one first sequence is allowed, and there will be validation for some parameter combinations as well. Once you submit the job, you will be getting the job ID, and which you can actually share with your colleagues. And you can also see the status of the job, whether it's queued. That means the analysis is not yet started, it's still in queuing. And once it's started, you can see it's running. Once it's done, the results will be displayed in the same window. And if you're searching for a job that is no longer available in our server, like you will be getting a job not available or a job not found as status. Okay, so this is how the result page looks like. This is an example of Clustal Omega result page. You will be having multiple tabs where you can navigate through. And a few of our applications also provide workflows. Say, for example, for a multiple sequence alignment tool, it allows you to launch the result, pass the result of this tool to another tool, like, say, for example, simple phylogeny or MView. We also have workflows in our sequence similarity search tools. From NCBI BLAST, you can launch MSAs of multiple sequence alignment from selected hits, and you can then see the phylogenetic tree from there. Okay, now let me just take you to the job dispatcher website. So this is how our new website looks like. So we have already covered these parts. So here you can see help and privacy. So it briefly explain what and what we are and how to get help from. This is the main documentation page, which is available from jdispatcher/docs. So this gives you a detailed info about all our tools and how to use programmatically and FAQs. So if you go here, you can see what are the available databases for our similarity search tools and a few example formats as well. If you are interested in programmatic access, you can come here and explore more about the programmatic, the Open API documentation is available from here. So if you choose a tool, you can actually go through this. I'm not explaining this programmatic access in today's webinar, but we have a policy in place for our programmatic access users that we request you to submit 30 jobs at a time in order to make a fair use policy. There are a few examples as well to run, how to use our clients, web service clients. There are frequently asked questions in general and also tool related. If you come here, like you can see, tool specific FAQs are also available. So that's all about our documentation. So, okay. So let me now show you how to launch a Clustal Omega tool. So Clustal Omega, it's a multiple sequence alignment tool. So it expects more than three or more than 3 sequences. So I am now providing this interleukin-2 receptor protein sequence from different organisms. You can see here, which is from UniProt. So I have specified, so by default it's protein. So this is a protein sequence. So I'm just, I have a file here, which is in FASTA format. So I'm just uploading it. And if you want to change the parameters, you can change it here. Like if you want the output in some other format other than Clustal W format, you can choose it. And there are also options to change and I'm now using the default parameters, and I've submitted it. So you can see that I got a job ID here. It's submitted and queued, and it's running now. It's finished, and here is the output. So this is the actual alignment that this tool produced. And you can see the conserved regions from here, like these are the conserved regions. So this is the actual alignment. And if you come down further, you can see the alignment with colors. This one, we have preserved the same format that the same color formats that we used to before, because we got a few user requests to preserve it. So if you wanted to know what these colors mean, you can always go to the FAQs, and it gives you what this color indicates. I mean, which group of amino acids indicates this color. We have recently added a new feature, which is a Nightingale view, which is from the UniProt. So you can see the alignment in one line. If you scroll through this bar, you could see the entire alignment in a row. So by default, it uses the Clustal 2 color combination. So the amino acids color codes are shown here. So if you wanted to change into serine and threonine, like you just wanted to see the serine and threonine residues, like you can choose this color scheme and see the alignment here. There is also option to zoom out and zoom in. Then comes the phylogenetic tree. So we are using this phylotree.js for generating for the tree construction. So this is the phylogenetic tree generated from this multiple sequence alignment. So you can increase the size, and there is also option to change the view. You can also select branches. So if you select a branch, you can see that here, like you can perform actions, delete, and you can also perform several other actions like collapse subtree or hide the subtree, and several other actions. The result viewers, this actually gives the workflow. So if you wanted to pass the result of this, this alignment to any other tools. So we have like MView and simple phylogeny, and JALView is also there. So JALView, you may need to install it. If you don't have installed, there are instructions available from our help page. So I have already installed JALView. So let me just open it, and I'll just show you how to provide the URL. Okay, just input alignment, and from the URL, you just specify this URL, and now you got the alignment. So this is a third-party tool that you are using to visualize that alignment. And there are also options like, to change the color, and several other options that the JALView provides. You can also send this result to another tool, say for example, MView. So you can see the job ID, which you got from here. So it's passing this job ID, and if you submit it, you will be getting the HTML markup color code added to this alignment. So let me just go back to, so this is the MView result, and you can download the result if you want. Okay, another tab is the result file. So here you will get all the results that are generated by this Clustal Omega tool. So this is the actual alignment. So if you want to download, you can download it. And okay, guide tree, which is not actually the phylogenetic tree, but it is used to generate the final multiple sequence alignment. So you can download the guide tree if you want, and the phylogenetic tree, the actual phylogenetic tree, and percentage identity metrics, and a few other files as well. The last submission details tabs gives you the version of the program, like the Clustal program, and when you launch the time you launch the job and finished, and the actual command you passed, and the parameters that you put for this analysis. So if you want to preserve these parameters later, you can always visit and take it from here. Next is BLAST demo. So let me just go to the job dispatcher homepage again. So BLAST comes under sequence similarity search. So if you click on here, it goes to the BLAST tool page. So first, select your sequence type. So here you can see these are protein databases. So you are trying to provide a protein, you're providing a protein sequence and trying to check for the protein and search against a protein sequence databases. If you want to search against nucleotide, you have a tab here. So if you come here, you can see that this is the nucleotide databases. And you can also search, like provide the nucleotide if it's nucleotide against nucleotide, which will be the BLAST N. But if you change this, it will automatically change to the T-BLAST N program. And I am using the example sequence here. So it's the human insulin protein. So from UniProt, and the program is BLAST P. So if you want to change the parameters, you can change the metrics or like other params as well. So once you submit, again, you'll be getting the job ID, and you can see the job is running. Okay. So this is how the result page looks like now. So you can see you have 50 hits found, because by default it's 50. You can change that in the parameter session. You can see the alignment option there. So the first sequence will be the sequence that you have provided, because it's again searching against the UniProt. And you have the length of the sequence, scores and percentage identities and e-values. We have newly introduced this feature where you can filter the result based on organism. So that is available. And if you want to view the annotation of a particular entry hit, you can actually check this in and can go through the annotation, like the feature table here. And similarly, if you want to see the alignment, so the query against this particular sequence, you can click on the alignment. You can select the hit and then click on alignment. It gives you that particular alignment, and you can also download the sequences in FASTA format. There is also a workflow available from the sequence similarity search result pages. So if you want to launch a Clustal Omega from here, say for example, if I wanted to align first maybe 6 hits, say for example, and I wanted to launch a Clustal, you can click on this and perform the alignment. Okay, the next tab is tool output. So this is the actual alignment file. So here you can see the scores and e-values, and coming down you can see the actual alignment for query and the sequence hits. Next tab is the visual output. So if you want to see the colored visual overview of the e-value, based on the e-value, you can get it from here. Like so here, if the e-value, like it's red for 0, and you can see that it's coming down like when you're going on the left hand side. So that is there. And the functional predictions, so you can also see the functional predictions from InterProScan. So you can check for that by clicking on it, and you can see the position and where it starts and ends, the residue positions as well. And similarly, it also got the result files, all the results files that are generated by this tool. For example, if you wanted to see the accessions and IDs. So this is IDs. So if you wanted to get all the IDs, you can download it from here. Sorry. Okay, this is the Clustal Omega result that we got after we launching a few 6 hits from the BLAST. We just close this. Okay, so so this is the identifiers, and this is for the accessions. So you can download it. And similarly, it has got like the submission details also, if you wanted to preserve it, or if you wanted to see what are the parameters that you have used for your analysis. Now, there are a few things that you may need to keep in mind, because occasionally memory-related problems may arise when the tool is unable to allocate enough memory to complete the task. If you encounter this issue while attempting to align extensive or lengthy sequences, consider using an alternative tool with similar functionality. Also, please note that our tools have file size limit, so ensure that your input falls within that range. If not, you can submit that as in chunks, like you can cut short the number of sequences and submit as like a different jobs. So I would like to highlight some of the advantages of using these tools in a job dispatcher. So we provide a user-friendly web interface, and most of these tools, say for example, Clustal Omega, that actually doesn't have any interface, and you may need to install them locally. But we provide an interface to access these tools easily. We also provide workflows that we already seen for a few of our tools. So it will be easy for you to navigate between tools, and we support various file formats that may not be available in the actual tool if you are going for a local installation. Then for our similarity search tools, you can search against various EBI databases where you don't need to install the tool, I mean, like BLAST or FASTA or databases separately. We will be taking care of the database update. So you will always have access to the latest sequence libraries. We also provide the programmatic way of accessing our tools using web services. A few of our few of EBI teams are using our web services. Say for example, Uniprot is using our BLAST. So you can see this job ID is it's the job dispatcher. So they are using our BLAST web service. Similarly, InterProScan, for InterProScan, the web service is they are using job dispatchers web service. Okay, so this is the documentation URL, which we already seen. And if you want to cite us, you can always cite us with this. This is the latest publication in NAR. So we are more than happy if you could cite us. If you're using our tools, like please cite us. And if you want to know the if you wanted to redirect to the actual tools publication, or like the binaries of the tools, like you can always find it at the bottom of the tool page. For example, if you are in Clustal Omega page, and before it's bottom of the Clustal Omega page, you can see not only Clustal Omega, every tools page you will be having this bio.tools record URL. So if you click on that, it will go to the corresponding tools bio.tools page, and from there you can see the corresponding publication and the binaries URL to the binaries. And if you want to contact us, so you can always contact us via the feedback form, or if you are reaching out to us from the EBI's support and feedback form, you can choose the job dispatcher services from the topic, and you can write your message. And please provide the job ID, and please let us know about where you are struggling, so it will be easy for us to understand your problem. And it's always good if you could check if the issue is with your file size or input size, or try using a different tool if it's related to that, if it resolves the problem. And also try to go through the error message if that helps. If not, you can always contact us. We will always be happy to assist you with your queries.
First of all, thank you, Nanda, for today's webinar, and thanks everyone for joining in today. There's another question: what tool can I use for transcription factor prediction?
Transcription factor prediction. So it's something like, you have some sequence and you wanted to try like similar ones, right? So maybe like to start with, maybe you can use BLAST, like, if you have the sequence, and you can search against the like whichever database you are interested in, and you can maybe start, with the job dispatcher tools, you can use any of the similarity search tool.
Okay, thank you. Can analysis results be saved online on website?
Yeah, so it's browser dependent. So I whatever I'm seeing here, whatever jobs I'm seeing here, that's the jobs that I submitted. So if I open an incognito window and I use 1 second, and if I go to job dispatcher, 1 second. Yeah, I won't be seeing anything, and it's browser dependent. So if you try opening another browser, you won't be seeing any of the jobs. So yes.
And just to add to that, so how long these results are saved?
7 days. Yeah, a week, 1 week.
1 week on the server side, but in the browser, it's important to remember that the results, so your recent jobs, we don't recognize users really, so it's up to your browser. So you notice you don't log into that website, so we don't know if it's you. So the results of your jobs are stored in your browser. So if you are trying to use a different browser, you will not see your results over there.
Thanks, Anya. So the question that we had about the cluster has been expanded now. So Authorwin could be used to produce single copy gene clusters that have only one physical location in the gene. Single copy genes can have orthologs in different species and are used as markers in various studies, which includes phylogenetic comparisons and species classification. I want to know if there's any tool that could do the same.
Okay, so I think this question is specifically related to the phylogenetic studies, right? So the one we have currently is the phylogeny tool, which uses the neighbor joining method and UPGMA. If you are like doing intensive study in phylogenetic, we recommend you using some other tool rather than our job dispatcher tools.
Okay, so new one is the sequence similarity important, or the percentage identity is important?
Percentage identity is like that gives you the pairwise, so how much these two sequences are identical. So from the alignment, like as a whole, like for example, from the Clustal alignment, you can see like where are the conserved areas. So it's different.
A new one has come in. What is minimum percentage cutoff of two protein sequence identity that works as the same function? So essentially, what should be the minimum percentage cutoff to say
Depends on your
have the same function?
need, right? So if you have I think Fabi is also typing, so
Yeah, it's a bit tricky. I mean, yeah. The problem is you could have proteins with very similar structure and function with very minimal sequence identity. So it's like there's no number you can actually use. Actually, you would need to look at structures. So yeah, difficult. No arbitrary number you can come up with will be good.
All right, so there are no more questions. So I would like to thank Nanda again for the webinar today, and like to thank Anya and Fabio for answering questions. And thanks everyone for joining in today.